This isn't my overhead, but I'm glad that it's here. I knew Peter, and I worked with him on NIH study section, and he extended the invitation for me to come here today, and I'm very sad that he isn't here. I thought I'd start out by telling you a little bit about Hauser Institute. This is a pixelated version. Hauser Institute is in Los Angeles. It's in the famous Ramparts district of Los Angeles. You might know about that. We are a private nonprofit research institute. There are about 160 or 170 members of the institute, and the departments are all concerned with auditory and speech processing issues, and they run the gamut from cell and molecular biology, children's auditory research, electrophysiology, electron microscopy, auditory implants and perception, clinical studies, histopathology, hearing aid research, and our own department, which is communication neuroscience. Parenthetically, Hauser Institute is where the cochlear implant was invented, and where more recently the brainstem implant was invented and has gone through clinical trials. So this is, from the perspective of auditory science, a fairly major center of research activities. Our own work involves the Hauser Institute site, and also linguistics at UCLA, and the imaging center at USC. We have a laboratory for testing deaf undergraduates at Cal State Northridge, and I have a collaboration at Caltech. So we're fairly widely organized, and I'll just put up here some of the names of the people who have been making contributions to our work. Because what I'm going to try to talk about is really more than any one person, or maybe more than any one department could possibly try to do. Basically, can you all see this? Okay. What we're interested in is what's the basis for the human ability to perceive linguistically relevant, that is, phonetic information in the physical speech signals. What are the processes involved in recognizing words? And what are the determinants of individual differences? Because humans don't all do these processes equally, and particularly in certain disease or impairment states, they become even more different from each other. Over the years, it seemed to me that a couple of hypotheses have emerged as being key to our work. One of them is what I call the modality-specificity hypothesis. And what this hypothesis says is that auditory versus visual modality-specific phonetic processing occurs in the human cortex. So what I'm saying is that speech is processed specifically as speech in through more than the auditory system, but it also specific phonetic processing takes place through the visual system. And the lexical projection hypothesis is that the consequences of this modality-specific processing are projected to the level of lexical processing. So it makes a difference whether you recognize a word that through your hearing or looking at somebody who's talking. I have a couple of boxologies here. Okay, all right. This, I think, correct me if I'm wrong, is the standard boxology. Do I have a pointer? Thank you. I think we generally imagine that in the case of auditory speech perception or visual speech perception, that is lip reading, there's some kind of a phonetic module that processes the physical signal and delivers the information to some kind of lexical or word representation. And that this phonetic module is a single module, and it's probably involved with the auditory system. The kind of view that has come to me over the years of studying lip reading and auditory speech perception is that probably the first model is wrong, that there is this specific optical phonetic processing separate from acoustic phonetic, but they do interact in ways that we're just beginning to understand, and I might have time to get around to that. So here's what I'm going to try to cover, and it's probably overly ambitious. The first thing I want to do is tell you about the phenomenon of visual speech perception and try to argue that looking at someone talk can be a sufficient basis for processing speech. Then I want to describe to you a computational study that starts to explain why that might be possible. That leads into a study to test the model, the computations, and then I'll look at some similarity relationships that we've studied between optical signals and visual speech perception. And finally, I'll try to get around to the cortical substrate for visual speech perception. If I have any time left, I'll try to talk about the complicating factor, which is that we have two senses and they interact. Yeah.
You're saying that as far as just the visual processing is concerned,
yes,
that what goes on in my cortex if I'm just looking around at trees is done by something which is more general, but if I'm looking at you as you speak, that part is doing something special because I know that it's speech.
Yes.
In other words, this is not
or your brain, that part of your brain knows that it's speech. You probably also have a part that's specialized for trees as opposed to faces too, but we won't get into that. Yes. But in the same sense that you have a part that's specialized for faces, you may have a part specialized for speech. All right. When I was at Hopkins working with Maurice Goldstein, we were interested in the problem of speech perception by people with profound congenital deafness. At that time, cochlear implants were a new thing, and they were controversial, much more so than today. We wanted to know whether we could put speech in through the skin as opposed to putting electrodes in the cochlea. But we knew we couldn't get it all the way via the skin, and that probably we'd like to combine signals applied to the skin with lip reading. So lip reading is something of a misnomer. It means looking at a person's face while they're talking. We did our experiments with undergraduates at Hopkins, many of them who are EE students, and they weren't real great at lip reading. So that seemed to suggest that people actually couldn't do that task very well. But then when I went on to Gallaudet, which is a place where all the undergraduates must be deaf, because it's a school for deaf students, we met people who seemed to do very well perceiving speech primarily through their eyes. So we decided it would be a good idea to find out what very well means And we conducted a large, we call it normative study, in which there were 72 deaf undergraduates and 96 hearing undergraduates. And we compared them on lip reading nonsense syllables like ba, da, ga, versus, in one set of trials, isolated monosyllabic words like book, car, in another set of trials, and then isolated sentences that don't relate to each other. And for this experiment, we're just showing them the talker and asking them to indicate what the talker said. I'm going to show you some box plots. What these are plots showing the entire distribution of the data. And here are the deaf results. We had a male and a female talker, so the red and clear are just the talkers. Here are the hearing results. And what the box plot shows you, the line through the center is the median, percent or proportion syllables correct for the nonsense syllables. This top whisker here is the fourth quartile. You can see here third quartile, second quartile, first quartile. And what you should take away from the slide is that indeed nonsense syllable perception is not particularly accurate by just looking at a person's face. But statistically, the deaf subjects were more accurate than the hearing. When we went to monosyllabic words, we had one talker, and here are the results. What's interesting about these results is, well, for one thing, performance is higher in terms of proportion correct. But it's also true that the fourth quartile of the deaf subjects is more accurate than the entire population of hearing people. And finally, looking at the phenomenon here, we can look at sentence scores. And here we have deaf and hearing people lip reading fairly simple sentences, although more complicated ones are in the same ballpark. And we see people who can perceive speech through their eyes at around 80% words correct, which is like approximately listening to speech in something like -3 to 0 dB signal to noise conditions. So to some people, looking at someone talk is approximately like listening to me in this environment with the AC on and not the best listening conditions. And these are people, among these people are ones who have congenital profound deafness. So it's not that they had their hearing and lost it. They learned speech and language through their eyes primarily. So this study suggested to us that we could get high levels of word recognition and comprehension associated with reduced phoneme accuracy. So remember, the phoneme scores were relatively low, but the isolated word and sentence scores were relatively high. Implication. Many people have drawn from results like this that the deaf people were filling in or using top-down or guessing processes. The alternative is that in order to recognize words, you actually don't need that much information. And the bottom-up perceptual processes are adequate or sufficient for recognizing words through vision. So we decided to look at this first through a computational method which is related to some of the studies that have been done in ASR, in which the lexicon is coarsely coded. And the way that this method works is that we need to get an estimate of the confusability of phonemes in the visual domain. So we ask people to do the phoneme identification task. We present ba, da, ga in isolation. Forced choice, we ask people to tell us what they think the talker said. Then we apply cluster analysis, which is a statistical technique to group responses that are confusable. These are used to define what we call phoneme equivalence classes. So for example, in this particular slide, we have B, P, and M as an equivalence class. If you look at somebody saying words like bat, pat, and mat from the front, the gestures of the lips look very much alike, and they're highly confused. We created an online lexical database that has about 30,000 words in them transcribed phonemically. And we applied these equivalence classes as transcription rules. So, for example, whenever B, P, or M appeared, it would be transcribed as a single element. And after you do this transcription, obviously some words that were different before now become equivalent. Okay, so just to give you an idea, if it isn't already clear about the transcription rules, when you do the perceptual, the experiment in the psych lab, you get stimulus response confusion matrices, and now you want to model them as a hierarchies. And for example, this says that E and I are very close in the visual perceptual space. And then you can set criteria for how large of the responses you want to comprise in a set, and you can make cuts through the hierarchy. And so we looked at these sets of phoneme equivalence classes. So on the one hand, if you couldn't tell between any of the phonemes, you'd have one equivalence class, and if you could tell all of them in English, you'd have about 40. Once you retranscribe the lexicon, you can apply certain statistics to it. And I'll just look at a couple that are really simple. One is a frequency-weighted percent words unique. So we're asking the very simple question, if you do this retranscription, how many of the words still have forms that are unique in the lexicon? And as I said, in this case, it's a lexicon of 30,000 words. And what I'm going to show you is the result with frequency weighting, and the frequency weighting comes from the Kuchera and Francis norms. So here's the curve. Here are the numbers of phoneme equivalence classes and the percent words unique. And this little arrow here is at 12 phoneme equivalence classes. So what that's saying is, if instead of having 40 phoneme distinctions in English, you can only distinguish 12 groups, you can recover over 50% of the words in English uniquely. I think this 12 is approximately equivalent to what an average hearing lip reader could do. So it's probably close to what anybody in this room, randomly selected, might be able to do. We imagine that our deaf lip readers can do better than 12 distinctions, and what I think the curve shows is that a few additional distinctions buys you a fairly large amount. We can also look at the frequency-weighted expected class size. That's the average number of words that any one word would be confused with. And that's kind of interesting, because what it says is that even with only 12 classes of segments in English, the number of words that on average that you would confuse any word with is about 4. And very quickly, you're getting down to having only one other word that's confusable. if we break these statistics down in terms of monosyllabic words versus multisyllabic words, it becomes clear very quickly that most of the ambiguity is coming from the monosyllabic words, and that multisyllabic words, because they're longer and have more opportunities for combinations of the segments, maintain most of their uniqueness, even with just the visual information. So I'll just throw up this expected class size, which is kind of the reciprocal of that. So this modeling study showed that there's a high level of word uniqueness or distinctiveness that can be obtained with optical phonetic stimuli. And what this implies is that if the visual speech perception system actually works like this, we ought to be able to use this kind of computational approach to predict the accuracy for identifying isolated spoken words by vision only. So we did the study, and in this study, we selected 3 types of words. We had words that were predicted to be unique to lip readers, words that had 2 to 6 possible confusions, and words that were in large lexical equivalence classes, that is, words that had lots and lots of possible words that had the same form. And we asked hearing and deaf people to look at someone saying these words and just tell us what the person said. We also had those words in each of the groups. We had monosyllabic and disyllabic words, and we had high frequency and low frequency words. And what the results show is that the predictions were correct. Words that were predicted to maintain their uniqueness to the visual perceiver generally maintained their uniqueness relative to words that had 1 or up to 5 actually potentially confusable words, and then perception was worse for the largest group. And the results are similar for deaf people. They're a little bit more accurate.
Quick question. I'm trying to imagine what those words might be like. I'm having a hard time thinking of a monosyllabic word which has no competition.
Which has no competition. I can give you one, I think. So the word bought. Okay, if you, B, P, and M look the same, but we don't have mott. It's not a word in English, and we don't have pot. So if you see, and you can lip read, it has to be bought. It can't be anything else. Good question. We also don't have bod, do we? Oh, we do. Well, that's a specialized one, you know. So
ignore bod because it's sort of a coined word.
Yeah. So, in fact, though, the statistics show that there aren't very many monosyllabic words that can be unique for the reason I think you're pointing out. There simply aren't enough elements to create uniqueness when there's such coarse coding.
And these are presented in isolation.
Yes. So the idea is that spoken language is driven by the data. And you have to recognize words in order to process sentences. So our view is that we need to look at the bottom-up process before attributing a lot of guessing and inferencing to this process. Okay. One other point. My colleague Ed Auer did a similar study using what's called the neighborhood activation model, which is a similar model to these lexical equivalence classes and phoneme equivalence classes, but it's graded. And he substituted the confusions for acoustic speech and noise to see whether the data from auditory speech perception and noise would predict these visual effects. And the answer is they don't. So this is a kind of first indication, along with the other modeling study, that there's something unique about the visual processing route. It has its own confusions, its own lexical patterns, and you can't just substitute the confusions that come from listening to speech, for example, and noise to account for the data. So we think that modality-specific phonetic information, that is the bottom-up stimulus, projects to the level of the lexicon. I wouldn't claim that this study proves it. It's consistent with it. We're trying to build a case on the basis of various kinds of evidence. Certainly, they could do so as they could not do so as abstract categories, which are by definition post-perceptual. It seems likely that the structures utilized by the perceptual system require an entirely different and novel set of descriptors. So it's like you have the physical stimulus and you have the linguistic categories. So that's one view. If that's true, then all this talk about phonetics, optical phonetics versus acoustic phonetics, wouldn't matter much, and my first model might be true. You could say that the lexical results that I presented were due to some kind of abstract coding. Well, yes?
That doesn't preclude having two different abstract.
That's true.
Yeah.
That's true. Okay, alternatively, phonetic categories are considered to be formed on the basis of data-driven statistical evidence reflected at the level of the nervous system. This is a coexisting view, and particularly nice work done in this area has been by Ken Stevens. He's written, for example, the hypothesis is that phonetic segments within a particular natural class have some acoustic property in common, and that the auditory system responds in a distinctive manner when this acoustic property is present in the sound. So there's a kind of transparent relationship between the data and the percept. And intervening there is in his case, the auditory system doing certain transformations. Okay, we hypothesize that visual phonetic categories share representations and processing mechanisms from perception. They're based on perception. So physical and perceptual similarity relationships were compared for visual speech. I'll give you much more detail on this. And a high correlation between the similarity space for the physical signals and the similarity space for the visual perception would be taken as evidence for a data-driven phonetic category. I think I'll digress here for a moment with this picture.
I just need to explain to us what is a physical similarity.
Yes, yeah. We have a large NSF grant, KDI grant, to study relationships between acoustic signals, optical signals, and mid-sagittal magnetometry signals. And this study that I'm about to present comes from that larger set of studies. And this side here shows the recordings in the mid-sagittal section of the vocal tract, and I'm not going to be talking about those. I just want to tell you that there's a database that has that in it. And in some cases, it also has these dots that were placed on the talker's face. So the talker was recorded with dots and these mid-sagittal recordings. But again, forget about this. What we're going to talk about is recordings where the talker's face filled the screen like this, and he had retroreflectors glued onto his face. And what we recorded synchronously with the video was the movement of these retroreflectors using 3 cameras, so that we could reconstruct in time the 3-dimensional movement of the face. And these are the proxies for the visual signal. So we're not doing analysis on the pixels. We're doing analysis on the physiologic measures in 3 dimensions of the talking face for these locations. All right. This study involved 4 Native American English talkers, and they were recorded saying 2 tokens of 69 consonant vowel syllables, where the vowel was ah, e, or oo, and the consonants were all the initial consonants of English. So we had a fairly large database of these materials. By the way, we also recorded, we have another set with the magnetometer and the outside markers and the acoustics and the video, which has been used in some other studies, which I didn't bring that along, and if someone's interested in it, I can point you to some results. So here's here are the markers, and as I said, we used a 3-camera system. It's manufactured by Qualisys, and we were sampling at 120 hertz in 3 dimensions off of the 17 markers. All right. Here's the plan. That's the study.
Well, those 3 in the forehead, are you leaving those out on purpose?
They were used to stabilize the head, actually. Okay, that's an interesting point. So when someone talks, they move their head around, and if you don't stabilize the head, then the movements on the articulators are additive with the head motion. So the ones on the right. So if I'm talking like this, it's going to look like these articulators are moving a lot. So the 3 here were used to stabilize the head in a 3-dimensional, with a three-dimensional space.
So the data has been sort of normalized for the head.
Yeah, but if you want the data unnormalized,
no,
you can go back.
Just check.
Yeah.
It's not that you need to do something.
Yeah.
For something like this, that looks near comparison.
Yeah, and in fact, we have data on prosody, where we've looked at eyebrow movement and that sort of thing. So it's all there in the data. For this purpose, it's normalized out. Okay, back to the plan. Okay, this study has 2 parts. Remember, the goal here is to show that visual speech perception is driven by the signal. Okay? So we need a way to relate perception to the signal. The way we're going to do this is we're going to do a visual perception experiment, and we're going to do physical measures. The visual perception experiment is like experiments I've talked about before. We have these nonsense syllables that I told you about. We bring someone in. They have to make a forced choice decision about which of the consonants they saw someone say. And we did that for the whole set of 4 talkers by 69 syllables by 2 repetitions. So that's like 10 hours for each subject to come in and mark and indicate their response. Then we use multidimensional scaling. on the obtained confusion matrices to construct a space in which, I think this is hard to see, but all the consonants are in this three-dimensional space. From that three-dimensional space, it's obvious that you can then take any two pairs of consonants and obtain a Euclidean distance. So then we end up with the Euclidean distances between all the consonant pairs. So we've taken care of the similarity relationships on the perceptual side. Now we need the physical measures. Let me take this off here for a moment. The physical measures were based on an analysis segment that began 30 milliseconds prior to where the acoustic onset was and extended for 280 milliseconds. Why 30 milliseconds before the acoustic onset? Because faces start to move before sounds come out of them very frequently. So the visual speech information is not exactly coterminous with the acoustic information.
Does it stay, if you were to track the two, does it stay on 30 milliseconds?
Does it stay?
If you were to track visual versus acoustic,
Yes.
do you see this consistent latency?
Yes. Well, what happens is if you're trying to edit tokens for your database, you say to yourself, oh, I know about acoustic speech. A syllable is 500 milliseconds. I can fit lots and lots of speech on my optical disc or whatever it is. And you find out you can't, because you need about a second in front to get the face from a neutral position through into talking, and then you need about a second, at least at the end, to get the face to quiet down again. So unless you're willing to start in some kind of strange position, which is already into the articulation, it tends to be longer than the acoustics for an isolated utterance. Okay, so here we are at the physical side. The technique then is we have these points on their individual channels in 3 dimensions, and we're going to compute the Euclidean distance between consonant pairs using these points in 3 dimensions across the, how many frames? For 280 milliseconds. And then, having acquired the Euclidean distances in a matrix for all the pairs, we need to relate the two. And the technique was multilinear regression. So the objective of the multilinear regression was to estimate the perceptual distances from the physical distances. Okay? So we're looking for a transformation matrix that allows us to predict these perceptual distances from the physical distances. And that results in 2 vectors that are 253, have 253 components. And we're going to do just a Pearson correlation on those to see how good the prediction is. So that's the technique. All right. So here's the multidimensional space. And by the way, here's the intelligibility for the talkers and the various vowel contexts. And you can see that it's approximately what we expect, around 30-some% correct for identifying these. Here are the Pearson correlations. Obviously, you can break these down in a lot of ways, and this is just according to the vowels and whether we're focused on just the lips, just the cheek markers, just the chin, or all of them. And I'm going to look at all of them. The Pearson correlations are really high, I think. Let's look at them in detail. All right. it's always good to look at scatter plots to see what your data really look like. So here we have the perceptual distances, and here we have the weighted physical distances, which are really the predicted perceptual distances, and this is our worst correlation. So this is for consonants followed by the vowel ah. It could be that that this correlation isn't so good because when someone, it's good, but it's not as good as it could be. And it could be that when someone produces a syllable with their mouth open as an ah, you can see into the mouth. And in this particular analysis, we're not taking any data from inside the mouth.
Each circle there is a pair of syllables.
I'm sorry, I can't hear you.
In that each circle there is a pair of syllables.
Yes, right.
Sorry, the pair of consonants.
Right. So each of these is a distance. So these are the perceptual distances, and these are the predicted perceptual distances. And here we have the consonant followed by E. And for me, as a person who works with behavioral data, that's a very pretty correlation. And best was the scatter plot for the consonant followed by oo. Isn't that the best? Yeah. It's possible that the reason why this is so good is, again, because of the vowel, that the vowel oo involves rounding the lips, and it produces some co-articulatory effects, and it seems like both of our analyses, that is the perceptual analysis and the optical analysis, are picking up on that information. For your interest, I did a regression analysis of the weights, of those transformation weights, and they're not very highly correlated with each other. So it's clear that to do this analysis, you have to have a different transformation matrix W for each vowel context. All right, what do we conclude from this? All right, visual perceptual similarity structure exists among English consonant phonemes, and it corresponds to the similarity structure in the optical data. I think these results are consistent with the hypothesis that the visual phonetic categories are driven by physical attributes of the stimuli. And this again implies some kind of modality-specific processing, which, if you think about it, has some implications also for audiovisual speech perception. I may or may not get to that. Okay, what's the substrate? Now we have to go into neuroscience. These are results from a science paper that was published in 1997 by Gemma Calvert and colleague. They were the first people to publish data on functional magnetic resonance imaging during lip reading. And their claim was that they found evidence that when people looked at people talking, there was activation in the primary auditory cortex. And their interpretation was that the way people processed visible speech was that there was a route from the occipital locations to the primary auditory cortex. Okay, specifically, they were looking at an overlap, I think here and here. We thought maybe they were right, because when a paper is published in Science, you know, even though you know how the papers get in there, you think they might be right. On the other hand, we're working with an auditory neuroanatomist. And she was skeptical because if you look at the neuroanatomy, it's not clear how visual information would get into primary auditory cortex.
Is it possible that these people are just trying to listen very hard even while they're looking because they know they're supposed to figure out sound?
Right, but primary auditory cortex is activated by a network that is efferent. So there shouldn't be information. It's direct from the cochlea. Okay, so I don't know of any evidence that there's optical, that the wiring diagram works this way. But I could be wrong about that. I'm not a neuroanatomist. So what we're interested in here is, this is a brain, and here's the back of the head, and here's the front of the head, and primary auditory cortex is in the transverse temporal gyrus. It's a small area. It's on Heschel's gyrus in here, and this is looking in the coronal plane, where this is the side of the head, this is the middle, this is the top, this is the bottom. We're looking through this slice. So it's important in looking at some of the results I'm going to show that you remember about this. Anyway, primary auditory cortex is not on the surface of the brain. It's on this plane that goes into the head about here. Okay, here's where we're going. Other results in brain imaging suggest that there's a flow of information for acoustic speech from inside, tucked inside here, primary auditory cortex, down along this lateral surface of the superior temporal gyrus into the superior temporal sulcus. The route from vision is along here. In order to look at this, we conducted a study using functional magnetic resonance imaging. And in this study, we had hearing people and deaf people. For the hearing people, in order to localize primary auditory cortex, we had a condition in which people listened to a pulse tone and contrasted that with silence. So we're localizing the anatomy functionally. And in a lip reading experiment, they viewed people, a person saying words, and they had a control task. Let me show you. Okay, so the lip reading stimuli were this talker saying isolated words. So this is animated. And the subject's task, while in the magnet, was to indicate whenever an isolated word was repeated. And we did that to keep them paying attention to the stimuli. And they're lying in the MRI machine, and there's a mirror, and they're able to see the stimuli projected on a back projection screen in the room with the MRI machine. The control task was this same talker, one still frame, using the same schedule as the words, and the repeats were these dots which were placed on the talker's nose, and the task was to say whenever the dots repeated in color. So in functional magnetic resonance imaging, you want to hold the task as much constant as possible and vary the stimulus. Unless you're interested in the task, but we're interested in the stimulus. Okay. Blah, 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 blah. about functional magnetic resonance imaging, okay? The way this is done is you have an active task and a baseline task, and then you make statistical parametric maps, and you look at on a voxel by voxel basis for whether they follow this pattern. All right, the main point here, we imaged our subjects initially in the transaxial plane, looking at this tissue. There were 4 slices in 10 millimeter contiguous sections. And we were trying to, particularly to see primary auditory cortex. Here are the results. All right. I'm showing you 7 sets of data. So each of these is an individual subject, and the activation is projected on the anatomy of the individual. So these are not normalized. These use the anatomy of the individual subject. And what's coded here is, in red is lip reading, activation to lip reading, yellow is activation to tones, and the blue is the common area. And I think you can see there's very little blue here. However, there was one subject that looked like they might be a candidate for visual information activating primary auditory cortex. But remember, these sections are 10 millimeters, they're 1 centimeter thick. And so there's some issue about the resolution of these data. So we brought the person back and had them do the task. And this time we imaged in the coronal plane. So top of the head, bottom, left, right. It's radiological. Here's the activation for the tones. Here's for the lip reading. Remember, I said that primary auditory cortex is on a plane, and here we see the superior temporal plane along here, and there's activation. But when we ask the subject to do the lip reading task, we don't see activation there. In fact, we see it in the superior temporal sulcus below. So it doesn't look, from our perspective, and we've done this in more than these subjects, like the route in to the word processing system is through the primary auditory cortex.
Are the 2 studies done in the same period of the boxcar?
Yes. So in order to get the co-registration, you have to get the subjects to lie there still in the MRI machine throughout the entire procedure. And then there is normalization that involves co-registration with the early images for any movement. And because we have their anatomy, that helps as well. But basically, the subject is told, lie down, get comfortable, we'll wedge your head in place, don't move. All right. Without going into the gory details, because I don't have time, we did this study in the coronal plane with deaf people. And I'll just show you an example. This is from the anatomical image, and these mark where the sections are. So we're looking from the back of the head to the front of the head. And this would cover primary auditory cortex, which would be seen anterior about here. This is superior temporal sulcus. We've never seen in a deaf person use of primary auditory cortex for visual information, for speech. Oh, in addition, what you might not have noticed is that if you put these slices together, what you end up seeing is from the back of the head to the front, kind of a band of activation that goes like this. Okay. So that's what's in this picture here. And the claim is, well, okay, that this along here includes some specialized processing for visual phonetic information, different from the processing along here, which is for acoustic phonetic information. Bear in mind, however, this has not been perfectly mapped out in either the acoustic or the optical domain. But we do think that we have evidence that primary auditory cortex is reserved for acoustic stimuli. So it's not the way that visual speech information gets dealt with. So that looks like that's evidence for that model 2 boxology I showed you, where there's an acoustic phonetic and an optical phonetic processing route. So far, findings in deaf and hearing people are consistent. So not being able to hear doesn't mean that the auditory system gets co-opted by the visual system. So the implication is, among other things, that there is this modality-specific processing of visual versus auditory information. Another implication, which I think is really interesting, is that speech processing is more widely distributed in the brain than we thought. Who would have thought that there might be some processes back here in the occipital lobe or in association cortex here that might be specialized for visual phonetic signals. Okay. Should I stop there? It's an hour.
I think the regular learn to 5:30, so we probably, that's natural point to stop. Probably
Okay.
we should end here. And
If anybody has a
I
question.
did have a question. Okay. You showed that one slide that you had the red, yellow, and blue fMRI activation. And I guess one of them was the 1 kilohertz tone that sort of was your reference for
Not a 1 kilohertz tone, a 1 kilohertz pulse tone.
pulse tone.
Yes, because it's a complex signal, right?
Right.
Now you go ahead.
Right, and my question was, so this overlays the lip reading and the reference audio. What if you did the same thing with lip reading and the reference visual, where you had the colored dots? Do you see a lot of overlap?
Okay, so we're running an experiment now. We use the dot patterns rendered into video of the speech. So we can either have speech in full video or speech as these point light displays. I also have non-speech. So I have full video speech, full video non-speech, like face motions, and optical tracks rendered in video for speech and for non-speech. These are not results you can eyeball. You have to specifically do the statistical parametric maps for the interaction effects. But looking at it, it seems that it makes a difference whether you're looking at the full video face versus the point light. It makes a difference whether you're looking at a face that's talking versus a face that's making faces. That's what I get from just looking at the images. I haven't had a chance to process the data yet.
Yeah, in fact, my question was not even that complicated.
Oh.
I was thinking of a visual task, which is clearly not lip reading.
Okay.
You had the dots on the green or red
Okay.
stars or whatever.
I have a different set of data for you. We did an experiment on fingerspelling. So fingerspelling is a system that some deaf people use to spell out, using their fingers, words. we were interested in this superior temporal sulcus area, which, by the way, along here, also seems to be activated for faces and for biological motion. So we asked the question, what would happen if we had people look at fingerspelling who knew fingerspelling versus who didn't? We did the experiment. The result was that for people who saw fingerspelling as gestures, and I'm simplifying this just a tad. So for hearing people, disregard these data. What we saw, just think of this as the anatomy of this person, was parietal activation to fingerspelling in the area that may be related to these mirror neurons. So if you see someone doing something, there seem to be some neurons that have to do with production and perception. But for the hearing people, we saw very little activation in superior temporal sulcus, whereas for the deaf people, we saw it. So the functional neuroanatomy depends not just on the stimulus, but what you know about the stimulus. So if the stimulus has linguistic significance to you, then it's processed differently than if it doesn't. Is that the answer to the question? Okay, yeah. Yeah, that's what we've seen there.